Good evening, all. Hello.
Good evening, sir.
Is it audible to you?
Yes, sir.
Right. Last class transcription process completed, right? Hello. In protein biosynthesis, we completed transcription. Protein synthesis, 2 stages, transcription and translation. Please be silent. 1 stage completed. That is the transcription. What is transcription? What is transcription? Are you there?
DNA information transmits in
DNA information transmit into the RNA,
mRNA.
which is carried out by which enzyme? RNA polymerase enzyme through 3 stages, that is initiation, elongation, termination, right?
Yes,
After
sir.
termination, mRNA strand prepared.
Yes, sir.
In prokaryote, which reaction takes place in cytosol, whereas in eukaryote, which cycle takes place inside the nucleus. After formation, the mRNA inside the nucleus next transport into the cytoplasm, where translation takes place. But in prokaryote, transcription translation takes place within the cytoplasm because there is no nuclear membrane to separate cytoplasm genomic material. That's why in prokaryote, transcription, translation, both are takes place inside the cytoplasm. Eukaryote, it is distinct presence of distinct nucleus. The transcription takes place in nucleus. As a result, formation mRNA, which is transported into the cytoplasm. Now mRNA ready to synthesize amino acids to form a protein strand, right? And last class itself we completed what are the requirement. What are the requirement for translation? After that, let's see the 1 by 1 stages in translation. First, what are the before requirement? What is translation? What is translation?
Process synthesis of proteins from mRNA.
Synthesis of
Proteins from mRNA.
right. Translation is the mechanism by which ribosome read the information of the mRNA, write corresponding sequences of amino acid linked by polypeptide bond. That is one amino acid to another amino acid between covalently bond by peptide to form polypeptide. This mechanism, this total process called as translation. Right? That is definition. And what are the requirement? Requirement of the
Ribosomes, tRNA, mRNA, amino acids.
right tRNA. In last class we completed tRNA structure also. Was it completed or not? tRNA structure.
Completed.
And ribosome, another one, ribosome also completed.
Yes, sir.
mRNA, also completed because through transcription mRNA formed, which is always triplet codon like this. Triplet codons, the form of triplet codon. For each triplet codon code for one amino acid, right? One amino acid, code for one amino acid. So like that, how many amino acids present in organisms? Total 20 different type of amino acids present. Right, okay, tRNA completed, ribosome completed, mRNA completed. And other one, transcription factors. Transcription factors also required. Sorry, translation factors. Translation factors also I will explain later what are the translation factors in prokaryote as well eukaryote. Right? These are the fundamental requirement to the translation. Definition clear, requirement clear. Let's see the stages. In stages, right. In translation, clearly 4 stages. First stage, activation of amino acid. Activation of amino acid. Actually, we can extend one another stages also. Second one, this activated amino acid transfer to the tRNA. Transfer to the tRNA. Activated, transfer to the activate. This activated amino acid called as amino
acid, sir.
Right. Amino acyl adenylate acid. Right. Which is transferred to the tRNA binding. tRNA. Transfer to tRNA binding. And right here the formation amino acyl tRNA complex. Form as a amino acyl tRNA complex. tRNA complex. What is second stage? Simply say formation of amino acyl tRNA complex. That is better. Second stage, formation of amino acyl tRNA complex. Right? What is third stage? This is very, very important one. Initiation. The most important one, initiation. Chain initiation or initiation. Initiation. Initiation what here? Initiation of
Translation.
Right. Initiation. Fourth,
Assembly of ribosomal subunits.
that is, these are all formed inside the initiation stages.
Okay, sir.
Okay.
Okay, sir.
Next, elongation. Elongation. Finally, termination. Final one, termination. Okay. 5 stages clear first. Hello? First, activation. Activation
Of amino acid.
of amino acid. First one, activation of amino acid. Second, amino acyl tRNA complex formation. Third, important one, initiation. Fourth, elongation and termination. Clear? Steps are clear?
Okay, sir.
Now first, how it is activated. Let's see the activation process. Actually, in cytoplasm, number of different 20 type of amino acids present in inactive state. This inactive state of different 20 different type of amino acids activated. Just wait. Activated by ATP, amino acid ATP. Here the enzyme involves synthetase enzyme with help of synthetase enzyme. Synthetase with help of synthetase enzyme formed amino acyl adenolic acid. Amino acyl adenolic acid and 1 phosphate separate. That means 3 converted into ADP. Triphosphate, 1 phosphate utilized, converted into ADP. Now amino acid activated because utilization of extra phosphate, automatically this amino acid activated, right? Now this activated, after this amino acid activated, this activated amino acid attached to the tRNA at 3 end. That means CC end. Already we learned last class the tRNA structure, where the 5 end, where the 3 end. 3 end CCA sequence are present. Okay? Clear? Hello?
Yes, sir. Yes, sir.
Yes, sir.
Right. At first step, ATP reacts with enzyme to form an enzyme amino acyl adenolic acid, right? In second step, what happened in second step? This amino acyl group is transferred from AMP to 3 end of tRNA. 3 end of tRNA. As a result, formation of amino acyl tRNA complex. Formation of amino acyl tRNA complex. Right? It's clear? Hello?
Okay, sir.
Right. Now, anyway, amino acyl tRNA complex also ready. That is the first step. That the first step means how amino acid activated, how tRNA, amino acyl tRNA complex formed. Now it is ready to bind with mRNA or bind with ribosomes, right? Now initiation started. After activation of amino acid and activated of tRNA, Now initiation stage started. Let's see the initiation of first prokaryotic translation, later eukaryotic translation, right? Screen here, but just wait. Are you able to see this?
Yes, sir.
Yes, sir.
Right. First in prokaryotic initiation. Right. So first, what is the starting codon to initiation? What is the starting codon for initiation in prokaryote as well as eukaryote? Hello? AUG.
Methionine, sir.
No, I am asking about MR, not about amino acid.
Codon.
mRNA starting codon.
AUG, sir.
AUG starting codon, not only in prokaryotic, in eukaryote also same AUG is the starting codon, right?
Yes, sir.
Now this AUG initiate the translation
Process.
The first let's see in prokaryote. Where are the translate, right? First 30S subunit. 30S subunit. 30S subunit first forms a complex with IF3, which then binds mRNA. Before bind, actually this diagram already binded to the mRNA. Before bind to the mRNA, first here what happened? Let's draw here. Are you able to see this?
Yes, sir.
Okay, right. So this is 30S. Small subunit 30S. First it forms as a complex. 30S subunit first form as a complex with IF3. Somewhere here. IF3. Initiation factor 3. Initiation factor 3 bind. Initiation factor 3 bind. Later, IF1 is added to the complex. Second, IF1. For suppose here, IF1. IF1 added to the complex. The function of IF3, actually, what is the nature of small subunit? Small subunit does not no longer free in cytoplasm. It is always try to bind with large subunit, right? To prevent binding of large subunit, to prevent it before the IF3 bind to the 30S ribosome. To the binding of this IF3, this large subunit does not bind to the small subunit. That is the first thing, right? Is it clear? Hello?
Yes, sir. Yes, sir.
Right. And next, what are remaining? IF2. IF2, initiation factor 2, with help of GTP, with help of GTP attached to the 30S ribosome, right? So later, after formation 30S complex, IF2, just wait, IF1, where it is, right. So now this complete 30S complex ready. The 30S complex first IF3, IF1, IF2 with help of GTP. Without GTP, it does not move, does not bind to the 30S ribosome. Right? Anyway, this complete complex formed. But another one is, the most important thing is, why only this 30S ribosome exactly attached at AUG region? Actually, it not directly attached at AUG region. Before AUG, some interesting sequences are present in ribosome. Already told you what are present, how the ribosome made up of ribosome. What are the components of ribosomes?
Two subunits are larger subunit, smaller subunit.
No. Of course, two larger and subunit, but these two subunits made up of
Mg plus 2 ions.
and
Mg plus 2 ions and some amount of
some amount of
chlorophyll, I think so.
we are talking about ribosomes.
Ribosomes.
Ribosome made up of
Proteins.
protein and
Proteins. Mg plus 2 ions and ribosomes.
another important thing. Proteins and rRNA.
Yes, sir. RRNA.
rRNA. In rRNA, 30S subunits, mainly 60S rRNA present. 60S rRNA present. 60S rRNA. So this 60S rRNA contain the special sequence. This special sequence called as here. We can observe here. This is mRNA special sequence here. The specialized sequence called as, actually not given in our book, but write down before attached to the AUG, first the 30S attached to the specialized sequence of 16S rRNA, the specialized sequence called as Shine-Garneau sequence. Shine-Garneau. Shine-Garneau sequence. Shine-Garneau. Here. Shine-Dalgarno, right. Shine-Dalgarno. Shine. This is very, very important and competitive. Dalgarno sequence. Whereas in eukaryote, Kozak sequence. In eukaryote, sorry. Prokaryote, Shine-Garneau. Eukaryote, Kozak. Sequence very, very important. Right? The RNA Shine-Garneau sequence may be combined with like U sequence U, C, C, U, C, C, A. This sequence, Shine-Garneau sequence U, C, C, U, C, C, A. So this sequence called as Shine-Garneau sequence, right? Whereas in Kozak sequence, eukaryote, Kozak sequence, which is T, Kozak eukaryote, where it is ribosome G, right? G, C, G, C, G, C. Almost similar. So this is Kozak sequence in eukaryote. But in prokaryote, Shine-Dalgarno sequence UCC, UCCA. After that, AUG sequence initiation codon present. So
Sir, if
first
you don't mind, can you please repeat it again, sir? What is Shankar? I didn't understand, sir.
Shine-Dalgarno is the first recognition site. Actually, the 30S ribosome first attached to the mRNA, right?
Yes, sir.
But here, but where exactly it attached? How can recognize the AUG sequence first?
In the Shine-Darko sequence, sir.
First, that's it. First, if it is identify the Shine-Dargarno sequence, just side of the Shine-Dargarno sequence, AUG codon present.
Okay, sir.
Right? That means actually what happened here? So this is U, C, C, U, C, C, A. This is rRNA sequence of Shine-Dalgarno. What about mRNA sequence?
A, G, G, A, G, G, U.
Right?
Yes, sir.
So that means here the mRNA, this sequence complementary to this sequence. Now, where it is exactly attached to each other. Right? After that, scan some place, this portion, later reach the AUG region.
Okay, sir.
After reach AUG region, what happened? What is the next step?
Next elongation, sir.
No.
Next second codon will be recognized and
First codon recognized. Does it recognize or not completely? Only we know AUG. But AUG bind with methionine or formal methionine?
formal methionine.
In prokaryote, formal methionine, eukaryote methionine.
Methionine. Okay.
Right? But does not bind it.
Okay, sir.
Actually, we started with first this complex formation. This is the first step. Don't get confused with the help of these steps. First steps, right? What happened in the second step? In second step, with the help of Shine-Dalgarno sequence, the 30S ribosome, mRNA, this mRNA bind to the 30S ribosome. Okay? mRNA stand, actually not 30S. Here, mRNA itself directly bind to the 30S ribosome, where exactly Shine-Dalgarno sequence present.
Okay, sir.
After that, now in next step, tRNA, amino acyl tRNA complex, with formal methionine, FMET, formal methionine reached the AUG sequence here. This is AUG sequence. The anticodon sequence should be UAC. It is complementary to the AUG sequence, and it is code for one amino acid, that is formal methionine. Right? This is also clear?
Yes, sir. Clear.
Now what remaining here? Now here 30S ribosome, okay, clear. GTP hydrolase, okay. Next IF1, IF2, IF3, decided ribosome are used again. Start chain, right? IF1, IF2 are required for binding of initiation for tRNA to 30S ribosome. Mainly IF1, initiation factor 1, initiation factor 2, These both are required for the binding of initiation of tRNA to 30S ribosome. Okay, these are all involved, IF1, IF2, IF3, but how the tRNA bind to the 30S ribosome? It's the tRNA with help of initiation factor 1, initiation factor 2. With help of this initiation factor, the tRNA bind to the 30S ribosome. Initiation factor 2 is involved in binding of guanine triphosphate, and IF3 is lysine arginine-rich basin protein control ability of the smaller subunit to bind mRNA. No need to read all this, just simple one word. Here the tRNA, let me draw here, it's tRNA, right? This tRNA with help of IF1 and 2. IF2 always with GTP, guanine triphosphate, bind to the tRNA and bring towards 30S ribosome. Bring towards 30S ribosomes. Here we can observe here GTP. 30S ribosome mainly here IF2 and IF1 also involved to bring tRNA towards 30S ribosome. Is it clear? First step complex. Second Shine-Darno sequence. In third step, tRNA with help of initiation factor 1 and 2 brought towards the 30S ribosome. Right? It's clear? Hello? Hello?
Yes, sir. Yes, sir. Clear, sir.
Any doubt, ask me.
No, sir.
Right.
Sir, in all prokaryotes, the Shine-Darno sequence is same?
Yes.
Okay, sir.
Right. Next. The remaining 50S subunit attached to the, that means it's completely closed. This for 50S closed the complex, tRNA complex. But here already, last class we discussed here 3 regions. P site,
A site,
A site,
E
E
site.
site. Here real same sequence also here E site. In small subunit also E site. Here P site, A site. Right? Now this 30S subunit closed this tRNA. Right? Sorry. Not closed it. Not closed, not covered the tRNA. In next codon, next tRNA also present. Next tRNA with help of same initiation factor 1 and 2, GTP energy attached to amino acid 2. For example, this is amino acid 1, considered as a 1. The second one is considered as amino acid 2. Now this tRNA amino acid brought towards A site. Right? Now 30S, 50S ribosome closed, completely closed it. So far clear? Hello?
Yes, sir. Clear, sir.
First site P site. Second amino acid A site. Third amino acid
E site, sir.
only first amino acid peptidyl site. Rest
Yes,
of the all coming amino acid attached at
A site.
A site shifted to P site. That is called translocation. That is called translocation. I will explain later what is translocation. Right? First, so far clear? Hello? Now this is initiation. Hello?
Yes, sir.
Yes, sir.
Now let's see the elongation process. And before that, just in rough eukaryotic initiation complex. In eukaryote initiation complex, different type of initiation factors are formed. Activation of amino acid clear. Activation of amino acid clear, but here just wait. Right. For initiation, different type of factors are involved. Instead of IF1, IF3, here 4F factors involved in eukaryote. 4F factors. 4F factors involved. Initiation factor 4F, initiation factor 4E, initiation factor 4A. Like this number of initiation factors involved. Right? And also here the formation of different type of complexes. First, formation of ternary complex. Next, formation of 43S initiation complex. Formation of 40S initiation complex. Formation of 80S complex. That is somewhat different when compared with prokaryote. I will give clear notes. I will give you clear notes during practical classes, okay, about the prokaryote and eukaryotic initiation. Right?
Okay, sir.
Right. Next
Okay,
one, in elongation. Initiation where the lung is right. Please be silent.
Hello?
Just wait, just wait. Here is elongation. This is mRNA strand, 5 end, 3 end. For suppose here maybe Shine Dargono sequence or Kozak sequence, here AUG. That is subunit. In elongation, in second step elongation, clear 3 steps are present. They are recruitment of binding incoming amino acid tRNA. Second elongation. First one, recruitment. That means binding. Just wait. Clearly. Are you able to see this diagram?
Yes, sir. Yes, sir.
Okay, right. So where it is. Full screen close. Full screen ఎట్లా రావట్లేదు ఇది ఓకే రైట్ in elongation first step incoming tRNA already told you incoming tRNA that is second tRNA second step peptide bond formation Peptide bond formation. Third step, translocation. This is very important. Translocation. Translocation. First, let me complete first one. Incoming tRNA. Incoming tRNA. Already tRNA present in P site, right? P site. This is A site. This is E site. Clear? Hello?
Yes, sir.
Yes, sir.
Right. Let me change color here. tRNA. Here amino acid 2. So that this is the incoming. Now next recruit the incoming tRNA. This incoming tRNA recruit where at A site. Recruit at A site. That's the first step completed. Clear? Hello?
Yes, sir.
Yes, sir.
Yes.
So the next amino acid enter the ribosome at A site as a amino acyl tRNA. Always the tRNA in the form of amino acyl tRNA. Amino acyl tRNA means here the activated mRNA, activated amino acid attached to tRNA, that is called amino acyl tRNA. Now this amino acyl tRNA and its anticodon pair with the codon of mRNA, and this facilitate with help of EFT. The second incoming tRNA brought towards the A site with help of initiation factor T. Sorry, elongation factor T. Elongation factor T in prokaryote or elongation EF1. This is in eukaryote. This is in eukaryote. EF1 in eukaryote, EFT in prokaryote. Mechanism same. What is the mechanism here? The incoming tRNA attached at A site. Okay. In association with GTP, and also here the GTP energy also required. GTP energy also required. From anyway, GTP, with help of GTP, elongation factor T in prokaryote, E elongation factor 1 in eukaryote, in both with help of GTP, upcoming tRNA attached at A site. Is it clear? Hello?
Yes, sir.
Now first step completed. Now second important step is peptide bond formation. Here the peptide bond formation between, where the peptide bond formation? Hello? Peptide bond formation between, peptide bond formation ekkada jaragalandi? First amino acid and second amino acid.
Second amino acid.
Right, peptide bond formation formed between first amino acid to the second amino acid, which is bonded by covalently. Right? Is it clear?
Yes, sir.
Hello? As a result, one molecule of water released. So in next step, formation of peptide bond between methionine. or first amino acid and the amino acid occupying A site, that means second amino acid, the peptide bond is covalently bond between two amino acid molecules. As a result, one water molecule lost. The initiator tRNA is released. That means first tRNA released. And this peptide bond formation, the carried out by the one special enzyme, that is peptidyl transferase enzyme, which is present in larger subunit. The peptidyl transferase enzyme present in larger subunit. Here the methionine first amino acids, the methionine residue is attached to the amino acids of A site by its carboxylic group. The simple, if you see the amino acid structure, let's wait. Let me draw here. Amino acid. Amino acid structure. Carboxylic group, here carboxylic group. Carboxylic group. First amino acid, for example, here. Right. CH3. H2N. COOH. This is one amino acid, first amino acid. Second amino acid, H2N, C, COH, something. This is second amino acid. So here the bond formation, first here the always one peptide bond, here loss one water molecule, right? Now this carboxyl group CO bond with amino group. This covalently bond, this bond, that means the share of the electron, sharing of electron. So it is called as covalently bond. As a result, loss of water molecule. This bond called as between carboxylic group of first amino acid, amino group of second amino acid. This bond called as, hello?
Peptide bond, sir.
Peptide bond formed.
Yes.
No, right. Now second stage also completed. Okay. First stage recruitment of tRNA. Recruitment of tRNA complete. Next, here the bond formation between second amino acid and first amino acid. Right? That is also completed. Okay, clear? Next, translocation. The translocation, simple here, the initiated tRNA P site, actually the first tRNA P site, it move to the exit site and exit away. Right? And this place, that means now A site tRNA shifted to P site, right? Hello?
Yes, sir.
Now the A site become empty. Okay? Hello?
Yes, sir.
Yes, sir.
This process called as translocation.
Dislocation.
Right, in this what happened, the mRNA peptide, after formation peptide bond, tRNA is transferred to the P site and mRNA with the next codon moves to the A site. That means P site RNA moves to the A site. This process called as translocation. In simple, in our convenience, just let me tell as a, here the first, this is A site, P site. The first P site tRNA move out. Next, the A site tRNA shifted to the P site. This is called translocation. This is called translocation, means just changing the places. Translocate. Translocate. The changing the, what are the changes here? P site E, P site become E, A site become P. Simple. Hello, this is mRNA. E, P, A. Okay. P to E, A to P. That is called translocation. Clear? Hello?
Yes, sir. Yes, sir.
Okay, sir.
In translocation also, it is carried out by EFG in eukaryote. Where it is? EFG translocator, right? EFG in bacteria. EFG for translocation involved elongation factor G in bacteria and elongation factor 2 in eukaryote. Elongation factor 2 in eukaryote. And here the utilize the GTP energy. In translocation also utilize GTP energy. right? Is it clear? Hello?
Sir, repeat again, sir. Once.
Where do you not understand? Hello?
Sir,
Simply explain the steps.
repeat again, sir.
Simple. Do you know translocation?
Yes, sir. Yes, sir.
Translocation shifted, changing of the places. E site A, sorry, P site E, P site shifted to E, and A site shifted to P.
Okay, sir.
Right? This
Yes,
is
sir.
translocation. This translocation carried out by special factors such as elongation factor G. This is elongation factor G. Elongation factor G in prokaryote. Elongation factor 2 in eukaryote. And this both are utilized GTP to translocate these places, to changes these places, utilize some energy and also some factors. If the factors are initiation factor G and initiation factor 2. Initiation factor G in prokaryote, initiation factor 2 in eukaryote. And also both are utilized GTP energy. Right?
Okay. Okay, sir.
Elongation also completed. Chain termination. Chain termination. In chain termination, right. Is it visible to you?
Yes, sir. Small.
Right. So anyway, always here we can observe, this is the first, this is first, are you follow this cursor? Hello?
Yes, sir.
This is first amino acid methionine, second amino acid, third, fourth, fifth, sixth, like this number of amino acids added by peptide bond. Always it move, this is P site, translocate E site, and this A site amino acid with tRNA shifted to the P site.
Right.
Always this A site become empty. This vacant A site always fill up with help of next tRNA amino acid. But when this A site, reach nonsense codon. What are the nonsense codon?
Opal, occur, amber, sir.
Right, U, A, G, R,
UGA,
U, A, A,
UGA.
other word UGA, sure? UGA. UGA, Opal, name.
Yes, sir.
These are another name, Opal, called as UAA, Ochre.
UGA, opal, sir. UAG, amber. Next,
UAG, this is amber, right?
amber, sir.
And UAA, ochre.
Ochre.
Right, when reach the A site. But these three nonsense codon, any nonsense codon occupied by A site does not code for any tRNA amino acid. Then utilize here the releasing factors 1, releasing factor 2, and releasing factor 3. These are called RF factors. 3 different factors attached to the amino strand or this ribosome subunits. When these 3 releasing factors, these are called releasing factor. After this releasing factor 1, releasing factor 2, and releasing factor 3 attached to this ribosome subunits, automatically this subunit dissociate, separated. and mRNA also separated. Now this polypeptide chain in the form of protein, this polypeptide chain, this in each polypeptide, it is built of number of amino acid, right? Now this completely formed as a protein chain. This protein chain also released out. So this is called translation. This process called as translation, right? Any doubt in this?
No, sir.
Clear initiation, elongation, termination.
Yes,
So
sir.
only in termination, one difference between prokaryote, eukaryote. In prokaryote, GTP energy not utilized for termination. GTP energy, GTP, guanine triphosphate does not utilized in prokaryote, but in eukaryote with help of GTP releasing factor. But here in prokaryote, 3 different releasing factor, but in eukaryote only releasing factor, one releasing factor present. That is the difference between prokaryote and eukaryote. Are you clear so far? Any doubt in this? Elongation process, termination process, initiation process. Hello? First here, the most confusing aspect is translocation. Translocation clear. Are you clear in translocation?
Yes, sir.
And another one is initiation. Actually, initiation's most complex in eukaryote. That's why I don't go for in detail. But I will give clear notes and time of practical exam. Okay?
Okay, sir. Okay, thank you, sir.
Right. Next. Translation clear. After that, next is what is the next in phytohormones, I think. Plant growth regulators. What are the plant growth regulators? What are the growth regulators?
Which regulates the plant growth, sir. Increase, they may increase or decrease the plant growth.
Increase or decrease. That's why it is called as regulators.
Plant.
Right. What are the regulators?
Auxins, gibberellins, cytokinins.
Right.
Auxins are for increasing the root. Gibberellins are for stem elongation. Cytokinins, they increase the dormancy period, I think so, sir.
Okay. Okay, right. No problem.
Auxins are for apical dominance.
Just wait. I will explain.
Okay, sir.
Are you able to see this diagram?
Yes, sir.
This is the auxin bioassay test. This is waste time, I think.
Coleoptile roots, sir.
Yes, coleoptile. Avena curvature test. That is whether the auxin present or not. To determine the whether auxin present or absent, Avena curvature test utilized. In this test was the coleoptile stem tip of the stem tip take and the auxin containing, let me take the color. Just follow my cursor. Auxin contain agar block one side here. Diffusion of auxins from the agar block. This waste time. I just can I directly go for physiological activity? Because again, this is repeated.
Yes, sir.
Yes, sir.
Right. Is it visible to you?
Yes,
Yes,
sir.
sir.
Right. So oxygen, first oxygen, indolestic acid. This is the common thing. Already you know the just introduction about the oxygen.
Natural oxygen, sir.
Yes, natural oxygen. The first discovered oxygen also
Indolistic.
IAA. So the bioassay is the Chalodny-Went theory. Chalodny-Went theory explain about the oxygen's existence. Just another important one, movement of oxygens. Oxygen movement in stem. The oxygen movement here, it is translocated through the phloem sieve tubes, not translocated phloem and xylem, but merely it is translocated from the parenchyma cells. The parenchyma cells of the xylem and phloem. Okay, only parenchyma cells. And this oxygen shows the polar movement. In short, the oxygen transport basipetal. Basipetal means, for example, this is the, are you able to see this diagram, my marker?
Yes, sir.
Yes, sir.
Okay, this is basipetal movement. Okay, clear? This is very important. In short, the oxygen shows basipetal. Basipetal means downward, right? As well in root, it shows acropetal. Acropetal means from root. This is shoot. This direction called as acropetal movement. So only here the mainly the movement of oxygen, polar movement. Polar may be acropetal or basipetal. Basipetal or acropetal movement. And the ratio of basipetal and acropetal movement 3:1. That means 3% basipetal movement and 1% acropetal movement. Was clear?
Yes, sir.
Right. Now let's see the physiological function. Physiological function, first one, phototropism. Second, gravitrophism. These three most important. First, let's see phototropism. What is photo?
Light.
Light. Trophism means movement. Toward the light movement.
Towards the
Toward the light movement. Simple. This is the stem. This is the light. You give one side this light. Actually, initially, here, are you follow cursor?
Yes, sir.
Are you follow cursor? Right. Here the oxygens are present. But when you apply with light, automatically these oxygens move toward the shade side, this side. These are all oxygens. But actually, initially, the oxygen present at complete portion. This complete portion oxygen, right? This apical oxygen shows the basipetal movement, downward movement. When you apply with the light, this oxygen from one side to reach another side. Another side. And where the movement of oxygens from this side? Hello?
Yes, sir.
Yes.
From this side, movement of oxygen goes upright or downright? Downward. Goes downward. Basipetal movement.
Downward, sir.
Basipetal movement.
But
While the oxygens goes down, the growth, for example, this is, let me bifurcate into two part, right? This is A part, light side B part. Now tell me, B part increased or A part increased?
8.3.
Right, A part increased, but not B part, because here the B part no oxygens. Already this, due to the light effect, this oxygen shifted to the one corner, A side corner, and where they are goes downward direction. Automatically this A side, this A portion shows the growth. As a result, bend towards light. Okay, is it clear?
yes, sir. Yes, sir.
So towards light. So this is called phototrophism. Direction of light. The plant growth, direction of light. So that is called as phototrophism. And next one, gravitrophism.
The
Gravitrophism.
experiment is the phototrophism.
Right here, we can observe here. This is the seedling plant, coleoptile, shoot tip, either root tip. Okay. Due to the gravitational force, oxygen reached the bottom, this place. Actually, initial, before gravitational force, this is completely oxygens, randomly distributed. Here also completely oxygens, randomly distributed, okay? For suppose there is no gravitational power appear like this, fill it with completely. But after gravitational force, what happened? Due to gravitational force, oxygen reached the bottom of the root, bottom of the shoot, we can observe here. Right? Right? Now tell me, this is shoot, right?
Yes.
This is oxygen place, okay? Now tell me the growth of shoot towards upright or downright?
Upright, sir.
Right, it goes this region only. This region goes upright direction. Like this. What about here? This is shoot. Sorry, this is root. Here oxygens are present. But in root, where the less oxygen present, only this region, this less oxygen region grows direction. As a result, shows downward movement. So both are opposite. It is positive gravitational. This is negative gravitational. It shows negative gravitational. It shows positive gravitational. Right? Here the oxygen growth increase. Less oxygen root increase. Clear? Hello?
Clear, sir.
Yes, sir.
Right. So one is complete. Sir, two, phototrophism and gravitrophism, both are completed. Now apical dominance. What is apical dominance? What is apical dominance? Apical, which is activate.
Plant length, sir.
Change difference.
So the growth which occurs at the tip of the plant part.
Right. Simple, the apical dominance means the growth, linear growth appeared when compared with lateral growth. That is called, generally plant grows more in length rather than width. This is formed due to the apical dominance. You see number of pinus plants. What happened here in plant? Are you able to see?
No,
Just wait.
sir.
No.
Just wait. Now clear?
Yes, sir.
Yes.
So auxin. Already told you basipetal movement, right? These are lateral bud. Lateral bud. Lateral bud. Lateral bud. Lateral bud. Lateral bud. What happens is, for example, this is 100% auxin. Now it is reached 90% auxin, 80% auxin, 70% auxin. So gradually decreased. Now what about this branches size? While the movement of basipetal movement gradually downward movement, for example here least, the least it is facilitate to formation of growth of the lateral branch. Second one somewhat small. Third one small. But ultimate growth is apical. Mainly focus on apical dominance. Right? But lateral, this lateral portions does not show much growth. Like this structures form as a cones. The plant formed as a cone. Right? This is called apical dominance. Another one is root formation. Root formation, the best one, IBA, indole butyric acid, naphthalene acetic acid. These two are synthetic axin. These two synthetic axins initiation of root from the stem. And another physiological activity, parthenocarpy. What is parthenocarpy? What is parthenocarpy? Parthenocarpy, fruit formation without fertilization.
Yes.
Actually, how the fruits are formed? How the fruits are formed? Hello? This is flower, androecium, ovule, female gamete. After fertilization, pollen grains reach to the ovule female gamete. As a result, this ovule, this entire body convert into fruit. Right? This fruit. But this ovule convert into seed. Ovule convert into seed, right? What happened in parthenocarpy? The male gamete, fusion of female gamete not occur. Directly, without fertilization, this ovary directly convert into fruits. Is there a chance to formation seed without fertilization? Is it possible?
No.
No, without fertilization, seeds does not form. That's why seedless fruits are formed. This is called parthenocarpy. This parthenocarpy induced by auxins. Now, sex determination. mainly in Cucumis cannabinus, it is induced female flowers, produced female flowers only. That is also effect of auxin. Now herbicidal activity 2,4-D and 2,4,5-T. These are act as a killers, dicotyledon plant killers, act as a herbicides. These are all auxin physiological activities. Right? Phototrophism, gravitrophism, apical dominance, and sex determination, root formation, and parthenocarpy. And simple code APS RTC. A P S R T C. Actually, here the C cell elongation, another one. Apical dominance, parthenocarpy, sex determination, root formation, trophical. That means what is this T? T for trophism, phototrophism, and gravitrophism, another one, and cell elongation also not given. Cell elongation and cell division, but cell elongation, cell division with help of cytokinin. The combination of cytokinin done this activity. So this is physiological activity of auxins. Right? Okay. Is it clear?
Yes,
Hello?
sir. Yes, sir.
Any doubt today class in protein synthesis? Please ask if you have any doubt. Hello? Hello?
Yes, sir.
Where a paper?
Can I stop?